Hi guys, thanks for checking out our video on mitosis and the cell cycle. Now, as we learned in class, mitosis, or the division of one cell into two daughter cells, is the ultimate goal and thereafter outcome of cell division, or better yet, the cell cycle. The cell cycle has four stages: growth 1, synthesis, growth 2, and M phase, in which a cell grows, prepares for division, and divides to form two daughter cells that then repeat the process. The phases from growth 1 through growth 2 are an interphase. Interphase is the period of the cell cycle between cell division in which the cell grows. The cell, though, must first tightly pack chromatin, a complex of DNA and protein, into a chromosome or chromosomes to ensure equal division of DNA when the cell divides. Chromosomes are thread-like structures within the nucleus that contains genetic information that is passed from one generation to the next. Once this genetic material is all packed down, the cell is ready to duplicate the DNA so that during mitosis, both daughter cells will share the same amount of DNA so that they can function properly. A singular chromosome will be made into two identical sister parts called chromatids, but the two sister parts will still be called one chromosome. The chromatids are connected by a centromere, the region of the chromosome where the sister chromatids attach, though it does not have to necessarily be in the center of the two chromatids. The cell will also duplicate organelles from the same reason that DNA is duplicated, to ensure that both cells have an equal amount of organelles to function properly. The centrioles will help to organize cell division in animal cells by moving to opposite poles of the cells, while plants organize spindles directly from the centromere regions and come into play later. Now for mitosis. The first phase of mitosis is prophase, the longest phase, when genetic material inside the nucleus condenses and the chromosomes become visible and the nuclear membrane dissolves. It is at this time that spindles, a fan-like system of microtubules that will help to separate duplicated chromosomes, start to form outside the nucleus and extend from where the centrioles are located. Next, in metaphase, the chromosomes line up across the center of the cell and spindles connect to the centromere of each chromosome to the two poles of the spindles. After comes anaphase, where the chromosomes separate and move along spindle fibers to opposite ends of the cell and require the disassembly of microtubules as the chromosome moves towards the poles of spindle fibers. Lastly, in telophase, the distinct and condensed individual chromosomes begin to spread out into a tangle of chromatin and the nuclear envelope reforms. Mitosis ends, but cell division does not. To conclude cell division, the cytoplasm divides to form two separate daughter cells in a process called cytokinesis. In animal cells, the cell membrane is drawn inward until the cytoplasm is pinched into two equal-ish parts. In plants, the cell wall is too rigid for this, and instead, a cell plate forms halfway between the divided nuclei, which then develops into cell membranes that separate the two daughter cells, and voila! The process is complete. Thank you so much for watching and good luck with mitosis.